The last part of 7.34, and I'm going to blend this into 735 and 736 because it's all about ribosomes. So 7.34 was the translation part, but they want us to talk about these things called polysomes. And basically, all a polysome is a polyribosome, which means a lot of ribosomes. And these a lot of ribosomes all line up on the same strand of messenger RNA, which means that they're all copying the information to create the same eventual protein, create that same polypeptide. And it's a very efficient way, especially if you need a lot of one type of protein. So I don't know if you remember, there was a video we saw where we were watching transcription and translation, and it said that we make 100 trillion molecules of hemoglobin per second. There's no way you could do that without many ribosomes reading the information off this messenger RNA, this single code. For example, if this code would be the only code, let me say that again, this code would be the code that allows us to create that molecule of hemoglobin, and we have polysomes to read it all at the same time and make many of them at once. It's a pretty cool image here where you can see all of the ribosomes here with all the little growing polypeptides coming off the tops and the bottoms. Pretty cool. So that's all a polysome is. The 7.3.5 just wants to know the difference, and to be honest, we've done this already, right? Know the difference between a free ribosome and a bound ribosome. So let's just talk about what the difference is between those ribosomes that are bound, which means connected, and those that are free. So free ribosomes are not connected. They can do whatever they want. They kind of float through the cytoplasm, and they make proteins that are specifically used for things within the cell. So all of these ribosomes are going to read the information off that messenger RNA to eventually make that protein that's going to be part of the inner workings of the cell. Oppositely, we have bound ribosomes, and here's one right here connected to our endoplasmic reticulum, and because they're on the endoplasmic reticulum, we call them rough endoplasmic reticulum. These proteins are going to be put inside this tiny vesicle, and then they're going to go to our Golgi apparatus, and then two things can happen here. So for these, they can be released outside of the cell, and this could either be a hormone, or perhaps it's going to be some sort of enzyme that's used in digestion outside of the cell. Or it could end up being something called a lysosome, I'll review, which are these vesicles, little bubbles with proteins in them that are used to help digest things within the cell. So these guys float around and they help digest. So that's the difference between a free ribosome, proteins outside the cell, bound ribosomes. They're going to be used for different processes outside the cell or for lysosomes. And the last little bit of information here is sometimes it's better to be a bacteria. I guess if it comes down to protein synthesis, I don't know. Don't quote me on that. But if we look at these two different cells, on the right-hand side, we have a eukaryote, right? And we know it's a eukaryote because it has a nucleus. Prokaryotes, bacteria, prokaryotes do not have a nucleus. So for them to make proteins, it's a lot quicker. It just can happen immediately after we do transcription. So let's look at this real quick because we know how it works in eukaryotes. We've been talking about it for the last week. Oh, look at that, polysomes down here. So here we have a circular piece of DNA. That's how DNA works in bacteria. And we're transcribing, if you can see it here, this little red piece, we're transcribing a piece of messenger RNA. And these ribosomes are just waiting outside for it. They don't have to be processed and introns and exons and then shipped out of the nucleus. No, we don't have to do that. They can just be right next to the genetic material, our DNA, and then it goes right to, here we got some polysomes, and you can make your proteins immediately. So that's all. It's just a state we just have to remember, but it's good to know the processes. However, IB does want you to be able to identify some. So in this image here, we have, this is a piece of messenger. Oh, no, this is our DNA. So we have a piece of DNA here, and there's it's hard to kind of see, but there's transcription happening here. And you've got these polysomes lined up here ready to take that information and make a make a polypeptide. Here's a little easier view to see that. We have our little messenger RNA there between our polysomes.